Okay, so forgot. So a warm afternoon to all. In the industrial microbiology lecture, today we'll start with unit number 3. Before that, we will complete the unit number 1, unit number 2, either on the fermentation designing as well as the basics of the industrial microbiology. Now the third unit, which deals with the scale-up process. What exactly that a scale-up process? In a layman language, it is nothing but the two important phases of the industrial microbiology. One we call them as the upstream process, and the second one we call them as the downstream process. And entire both the process we call them as the scale-up process. This is in the general way. What this particularly the upstream processing or the upstream process, what exactly and where the downstream process in an industry, it is very important. If we do not know the knowledge, then it is very, very difficult one how to proceed for any kind of the manufacturing of the interested product with the help of the microorganism. The upstream process, which having the clear-cut ideas of your product. As an industrial microbiologist, as a quality control officer, I know very well which type of the product is there and how that product is get forms, and how we have to go for the harvesting process. You have the ideas which type of this my product, whether it is a carbohydrate, whether it is a lipid, whether it is and the certain another things are there, whether it will be the primary metabolites are there, whether it will be the secondary metabolites are there. It is a different task. You must having the certain knowledge which is very, very important. Selection of the producing organism is very, very important. How to select the microorganism is a basic task in every process. Later on, when you selecting it, it is your moral duty. Go for the screening activity for the microorganism. That screening activity through the primary screening as well as the secondary screening. And after you know the screening, which type of the what you call them as the properties you are get a selected one, whether you are interested in the antibiotic, whether you are particularly interested in the acid, whether you are interested in the base one, whether there will be the addition of the indicators or not, which type of this product, whether already it is get mentioned in the certain protocols or it will be the new one with the help of the secondary. And whenever you are go for the secondary, how much amount of the product which can get released by the microorganism, you have to get the certain knowledge. Later on, you have to go for the formulation media. You have to designing the media as per your requirement. Which type of need is there? Whether it is for the pilot fermentation, whether it is for the large scale fermentation, that will be the industrial mass production. According to that, you have to formulate your own medias, because you have to add the certain waste material which containing the large amount of the carbon, nitrogen, it containing the minerals, it containing the vitamins, everything you must know. And later on, you have to optimize your media also. Okay, later on, there is the small scale bioreactor. We call them as the fermenter. We know very well which type of these fermenters are there, whether it will be the batch fermenters, whether it will be the fed batch fermenter, or whether it will be the continuous fermenter. You know very well. In a previous lectures, we are discussing about all these types of the fermenters are there. Aerobic fermenter is there, anaerobic fermenters are there, etc. and etc. Whether it will be the submerged fermentation, whether it will be the surface fermentations are there. Okay. And later on, if we know all these parameters, one, it is your main task. You have to show your guts. The process control a requirement in the form of the temperature, in the form of the pH is there, oxygen supplement, in the form of the carbon dioxide is there. Everything you have to keep in mind when you are proceeding with the control parameters one. And large, there will be the scale up of the process. So more than the hundreds of the liters of the fermentation mediums are there. But when you are selecting this one, you have to always keep in mind there are the certain kinetic reactions are there, whether it may be the zero order reaction, whether it may be the first order reaction, it may be the second order reactions are there, because that reactions play the important role. But at the same time, when you are doing the what you call them as the screening phenomena, we have to apply your own knowledge. For the strain improvements are there through the protoplast fusion techniques are there, recombinant DNA technology is there, okay, etc. and etc. So we call them as the molecular level things. Okay, and all these things a parameter merged together for the productivity, for the harvesting process, for the purification purposes one. That unit, it is always get attaches with your last and important phase or the processing. We call them as in the downstream processing. So everything, both this merging of the upstream as well as the downstream, we call them as in the scale-up process. Okay. Now the very first part, the isolation of microorganism. I will use the microorganism. Which type of the microorganism are you using for the industrial production? It is an ordinary microorganism where this particularly the microorganisms are available. Are you cultivated? Are you isolated in a pure form? Or are you buying from the market or the established organization where they are supplied the certain kinds of the microorganism in a lyophilized way? The organism should able to produce a higher yield of the desired product in which you are interested in. This is my first aim. When I selecting the microorganism, for what purposes I will select? I will select only for the desired product which can use the maximum yield. This is my first motto. If you are not thinking in that direction, you do not get the ample amount of the product. And if you do not get the ample of the product one, it is very, very difficult in a present scenario to establish in the market, because there are the large number of the players who can produce such type of the product. But every product having the different range. You have to put into that particularly the phase one. It is a very essential as an the economical way also. If your product will be the costly, nobody buy from the market. So the large number of the players already get playing their job in the market. Why they are interested? For your product. Already your product having the maximum price. So this is my first aim. Second important, when you are selecting the microorganism, always your microorganism should be in a pure form. They always be free from the viruses. They are always be free from the phages. So the alternate name to the viruses, we call them as the phage. Most important, your microorganism is genetically stable.
थांबणं 2च वाजेपर्यंत आहे. 1:30 वाजलंच आहे ना थांबणं.
हो 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 2 वाजेपर्यंत ज्याला घरी जायचं असेल तर लवकर जा, आपले मायक्रोफोन्स बंद करा. Bad manners, not having the proper etiquettes, someone delivering the lecture. If you are not interested, don't join the classes one. You are disturbing the system. Take the class on the online. So when you are go for the isolation purposes, you have the certain desired characteristic in my mind. My organism is 100% genetically stable. Means their genetic characters cannot get changes one. But when you are interested in the certain kinds of the mutagenesis process, your organism should be able undergo for the genetic modification. With the help of one of the best process, we call them as the mutagenesis. Your organism produces both the vegetative cells as well as the spore bearer. This is very important. If they are producing the vegetative cells, we are not in a position, all the cell having the same age, all the same cell having the same size, we are not in a position. But whenever there will be the spores are there, 100% when we are growing on the certain media, all the spores, they are get converted into the vegetative cells, and their size will be the same, their age will be the same. That is the main reason. Your microorganism grow vigorously after inoculation in the medium. भोयर भाई घरी जा आता. मोनाली भोयर. Okay. Now these students have do not proper etiquettes when they disturbing. They are not being online students. How they behave in a virtual class. If the same scenario is happening one, next time I will not sharing the my link. Okay. Now another important. Produce a single valuable product and no toxic byproducts are there. It is very essential. You must know the biochemistry. We must know the metabolism. If your organism gives a large amount of the toxic byproduct, there is no use. Product should be produced within a short time, and risk of the contamination should be a minimum under the optimum condition, because optimum condition, environment parameters are there, carbon source is there, nitrogen source is there, temperature is there, oxygen requirement is there, etc. and etc. That's why I'm using the word the optimum condition, but the risk contamination should be a minimum, or it should not get contaminates. Oh. Then what are the general methods for the isolation? We know very well as a microbiologist, we have the n numbers of the isolation techniques. The very popular method, it is a stick plate method. Sometimes we call them as the radiant, continuous, discontinuous, quadrant methods. Okay. So either it may be the central sticking is there, and most of the probably when you are going for the isolation, most of the times you have to always use the quadrant techniques. Another important method, that is through the pore plate techniques, where we can get the exact titer value. The hyphae techniques, this is generally used for the fungi. I'm not used for the bacterial purposes. For the bacterial purposes, it may be the stick plate, it may be the pour plate, it may be the spread plate techniques are there. But what about the fungi? Fungi can get produced with the help of the hyphal tip techniques. And the last but not least, that is on the macro manipulator techniques. We call them as on the single cell isolation technique. It is one of the sophisticated technique is there. Okay, so it required the highly sophisticated equipments one, but it required the high skill person. So you must know each and every the techniques having the proper principle. There are particularly the advantages as well as the disadvantages are there. Now this is one of the way how to isolate by the quadrant streaking. We call them as the four-way streaking. The first streaking, second streaking, third streaking, as well as the fourth streaking is there. So with the help of the inoculating a needle, so you have to make the first streaking over the surface of the nutrient agar. Again, the loop is get a sterilized one. Make the second streaking at the angle of 45. Go in a sequential manner, but the fourth as well as the first streaking should not get overlap. And now that plate you have to put into the incubator. Okay. At 37 degrees Celsius, if there is an what you call them as an the bacteria is there, but whenever there will be the fungi, so you have to incubate the plates, but at that time you have to do not use such type of the sticking techniques. Okay, so there are the certain different ways are there. Okay, so this is in the continuous one. We call them as in the central streaking. Okay, right from the top of the corner toward the another top of the corner of the plate one. So that is known as the continuous streaking. So this is your spread plate isolation technique is there. So all these plates are there. Plate number 1, plate number 2, plate number 3, as well as the plate number 4. In all these 4 plates, these are the nutrient agar plate already. Okay. Now, later on, with the help of the spreader, you have to spray this inoculum, which containing the microorganism. Okay. So those particularly the plate having the least dilution where the maximum amount of the microorganism, and those particularly the plate having the least dilution, there will be the well isolated colonies you observed one after the incubation period get over. So it may be the 24 hours, it may be the 48 hours depending upon which type of the bacterial strain is there. Okay, so this is the isolation by the micro manipulator techniques. So it required the highly sophisticated. This is one of the microscopic needle is there. Okay, this is your microscopic field where these are the large number of the microorganisms are there. So these are the prokaryotic cells are there. Now these particularly the microscopic needle, it is get linked with the a certain pressure. Okay, so you have to particularly press out the pressure and suck this one. Because of the sucking, only one of the microorganism they are get enters into the needle. Okay, so this is known as the movement of the field because of the pressure. So from one end, the observer who can observe the particularly the field, from the another hand, you have to just press the needle, and in the form of the suction, you have to give the certain pressure. Because of the suction, microorganism, 1 or 2 microorganisms, they are get just enter. That's why it is known as the single cell isolation techniques, or you call them as in the micro manipulator isolation techniques. If you do not take the much hard work for the isolation, and your industry do not permit the maximum time only on the isolation task, so there are the alternatives away. The microorganism can be obtained in a pure form from the recognized organization which maintain the cultures. The very first one, this is known as the ATCC. What is that ATCC? This is an American type culture collection, which is situated in Maryland, USA. Second most common, the Commonwealth Mycological Institute, CMI, mycological related to the fungus, related to the fungi, which is situated in Kew, United Kingdom. Third one, the Fermentation Research Institute, which is situated in Japan. Most important, as an Indian, the National Collection of Industrial Microorganisms, most popularly we call them as the NCL, National Chemical Laboratory, situated in Pune, Maharashtra. Another important, the Northern Regional Research Laboratory, USSR. This is in Russia. NRRL. And another very important, those particularly the microbiologists, they are interested in the production of antibiotics. They have to buy such type of the cultures from the USSR Research Institute of Antibiotics, which is situated in Moscow. So these are the very few recognized organization where they are providing the culture depending upon your orders. If it is an industry, so their charges will be different. Whether it is required for the student research activity, their charges will be the different one. So the cost of one ampoule, lyophilized ampoule of the culture, starting from 400, and the maximum for the industry, it should be a 50,000 to the 1 lakhs. So this is the organization who can provide it the what you call them as in the cultures. Now the screening of microorganisms. Already we are discussing these topics, but this is a part of the scale of the process. The primary screening, which associated with the 4 important techniques are there. The crowded plate techniques, oxygenography technique is there, enrichment culture technique is there by using the certain types of the indicator dye, as well as the secondary screening. Now, when you are screen out the certain microorganism, isolated the microorganisms from the nature, now it is your moral duty to preserve this culture. We call them as in the maintenance of isolates. And these maintenance of isolated by only the 6 important methods. We know very well the preservation methods for the microorganism in a laboratory. So it is always by the low temperature, you call them as in the slant culturing. The temperature that will be the 2 to 8 degrees Celsius, and it is always get kept into the refrigerator whose temperature will be the range of 2 to 8 degrees Celsius. Second, the frozen cultures. You have to frozen out the culture, remove the water molecules, and this is through the temperature -20 to -70 degrees Celsius. Another very important, the lyophilized culture with the help of the lyophilizer. The cost of one lyophilizer in the range of 10 lakhs to the 20 lakhs. We require the cold acetone. So whatever may be the culture which containing the water molecules inside the cytoplasm, that water molecules slow by you have to remove out. And your culture is get lyophilized and it can get stored years and years. Do not change in their genetic stability. Another best way, there are the certain gene banks. There are certain the what you call them as in the sperm banks are there. There particularly the cultures, they are get preserved in the liquid nitrogen. So there are the 2 types one. In a liquid nitrogen, one will be the liquid nitrogen and another will be the vapor nitrogens are there. The temperature of the liquid nitrogen is only -196 degrees Celsius is there. And the temperature of the vapor nitrogen is only minus 163 degree Celsius. You are go beyond the minus scale, and that culture can be preserved for a longer period of time. Nowadays, this liquid nitrogen, it is an very important as a medical services one. Most of the time, after the birth, their umbilical cord, they are get preserved in the liquid nitrogen for 10 years, 20 years, up till to the 25 years. And this particularly the institute, they can charges for a single year for the maintenance of the umbilical cord is only 25,000 rupees, and multiplied by 25 years. See the cost. Another important, that is on the air dried at a room temperature on a sterile sand as well as the natural substrate one. The last one, but not least, with the help of the recombinant strain, we are using the large amount of the recombinant microorganism. And these recombinant microorganism you have to preserve with the glycerin stab. So 0.85 ml of the glycerin and 0.15 ml of the inoculum mixed together and storage into the certain ampoule shell. So these are the certain parts one. Okay, so today I will stop here.